2020, and we're just starting our new lecture unit, Unit 7 on viruses, affectionately known as trouble wrapped in protein. So with the coronavirus pandemic, we know they are full of trouble. So again, folks, I'll try to keep these videos shorter. So we'll do several videos for this Unit 7 virus introduction PowerPoint. And then I'm going to try to work on a PowerPoint just on the novel coronaviruses. Okay. All right. So let's see if we can get this going. All right. So again, folks, one of the best descriptions of viruses are trouble wrapped in protein. So we'll do a little bit of history. Viruses have had a huge impact on human history. So starting off with just what we're experiencing right now, they've caused some worldwide epidemics known as pandemics. Now, fortunately, this novel coronavirus doesn't have the mortality rate of, say, smallpox, right? And it has lower mortality rate than some specific types of influenza. For example, the novel coronavirus doesn't have the really high mortality we see with avian influenza, the H5N1. But as we are all experiencing right now, viruses have been able to cause these worldwide epidemics and have caused a lot of suffering, directly and indirectly. Back in the 1800s, there were some scientists who hypothesized, that is, they made an educated guess, that there could be microbes smaller than bacteria, but they didn't know what those microbes were. For example, they thought that maybe there was a microbe causing polio or microbes causing smallpox, but they didn't know what that mystery microbe would be. Over here, folks, these are just examples of different hosts being infected by viruses. This is tobacco leaf infected with a tobacco mosaic virus. And this is a electron micrograph of E. coli, this little purple guy here, and it's being attacked by all these bacteriophage, bacterial viruses. This is, I believe this is influenza virus escaping from a host cell. Okay, so this is electron micrograph. And gosh, you guys over here, I'm forgetting what this one is. This looks like a mucous membrane, and these would be the little viruses here. So apologies for not remembering the caption on that fourth one. But what we do want to remember is that if you're a cellular organism, there's a virus somewhere in the world that can infect you. So with regard to virus discovery, viruses are so much smaller than bacteria, we can't visualize most of them with our light microscopes. So this is why it was so hard trying to find out what were these viruses. Was there truly some infectious agent smaller than a bacterium? So the first evidence was in the late 1800s. This is when the Russian scientist Dmitry Ivanovsky carried out this absolutely classic experiment. So people were really worried about a disease of tobacco plants. And tobacco, we know historically, and it still is, it's economically really important. It's used in making cigarettes and um, um, cigars, and people smoke it in pipes, right? So any disease of tobacco plants would cause, economic hardship on the tobacco farmers. And one such disease was called tobacco mosaic disease. And the reason it's called mosaic is you can see over here, see this nice, healthy green tobacco leaf. And then over here is a leaf from a diseased plant. So you can see all these little yellow, almost looks like little mosaic tiles embedded in the rich green of the healthier plant. So this is called tobacco mosaic disease. And Ivanowski was trying to figure out what caused it. So one hypothesis was, well, maybe it's caused by a bacterium. So he performed this beautiful experiment. He took some of the diseased leaves and ground them up in some saline. And then he passed that gamish or let it be filtered by these cool porcelain filters that had been developed by the Pasteur Institute in France, named after Louis Pasteur. And these porcelain filters, the pores were so small, bacteria could not pass through the pores, through the little holes. So if you use a porcelain filter, the filtrate, what would pass through the pores, would be free of bacteria, right? Because the bacteria were too big to pass through the pores. So Ivanowski took his filtrate of the diseased tobacco leaf, the filtrate which would have lacked bacteria, and he used that filtrate to inoculate a healthy tobacco plant. Now, if his hypothesis was that bacteria cause tobacco mosaic disease, what would be your prediction of what should have happened when Ivanowski used the filtrate to inoculate the healthy plant? Well, if the hypothesis that bacteria were causing tobacco mosaic disease was correct, the prediction would be the healthy plant should not get infected, right? Because the filtrate didn't have any bacteria in it. But what do you think were his observations? What happened? The healthy plant also developed tobacco mosaic disease. So Ivanowski once again had the prepared mind to kind of think outside the box. So Ivanowski explained that there was some filterable agent that caused the tobacco mosaic disease. So we hadn't um, developed the term virus yet. He said a filterable agent that caused the tobacco mosaic disease, and knew that it was going to be, sorry, our welcoming committee, our dogs are welcoming somebody at our door, that the filterable agent was going to be smaller than a bacterium. Okay, so then and sorry you guys for the background noise. So then a few years later, a Dutch botanist, Martinus Bijgerink, repeated Ivanowski's experiment. And this is always really important in science. If you come up with a new discovery, somebody else has to repeat your experiment to get the same results. Otherwise, you might be fibbing or, I don't know, making things up. And what was so cool was Bijgerink repeated Ivanowski's experiment, got the exact same results, right? And so it was Bijgerink that coined the term filterable virus. So here is our use of the term virus. And virus comes from Latin for poison. And Bijgerink described the filtrate as a contagious living fluid. Contagious, some pathogen transferred from one living host to another. But again, folks, they had no idea what it was. All they knew was the virus was smaller than a bacterium, and it could cause infection. It could cause disease in plants. And then if we jump forward to World War I, which was 1914 to 1918, um, there was a Canadian bacteriologist, microbiologist, who was working with the cavalry, the troops that still rode on horseback going into battle. And there was a huge problem because the cavalry were having an outbreak of diarrheal disease. And you can imagine that that would really reduce their fighting capability, right? If you're going into war on horseback and you have raging diarrhea. So what he did was he collected feces from the soldiers afflicted with the diarrhea. And just like you guys would do, he took the fecal samples and he inoculated the surface of agar plates. And as you guys can imagine, there's so many bacteria in feces. After incubating the plates, the colonies all grew together. So this forms what's called a lawn of bacteria. It's like you frosted the surface of your agar plate with bacteria, right? When the colonies grow, the colonies all touch one another, so we refer to that as confluent growth. Now, after incubation, the microbiologist held the plates up to the light, just like you guys would do. And what he found fascinating was there were all these hundreds and hundreds of tiny little holes in the lawn of bacteria. And these holes we now know were caused by bacterial viruses, viruses that can kill the bacteria they infect. And these little holes on the lawn of bacteria, they're referred to as plaques, Plaqueus, right? And we now know, again, they're caused by bacterial viruses, bacteriophage, and phage means to eat, so bacterial eaters that were infecting and lysing the bacterium. So this was another experiment showing the presence of viruses, right? But again, we still didn't know what they were. And again, folks, I don't know French. The Herr de Herrel, he again had one of these incredibly prepared minds, and he was thinking, what if we could use these pathogens, these bacteriophage, these bacteria eaters, to try to kill bacterial pathogens infecting humans? So he came up with this great concept of using phage is therapeutic agents, right? Phage to kill pathogenic bacteria. And again, folks, this was before the discovery of antibiotics, right? So pretty amazing. He also came up with a kind of a natural history, a life history of the bacteriophage, bacteria eaters, that they would attach to a bacterium, they would infect it, they would cause the bacteria to lyse, and then they would attach to a neighboring bacterium, infect it, and cause it to lyse. And through the successive rounds of infecting and lysing, that's what caused the plaques in the lawn of bacteria. So again, we're gonna go forward in time, folks, here to the Bay Area, where at UC Berkeley, the biochemist Wendell Stanley in 1935 was able to crystallize a tobacco mosaic pathogen. And this was really bizarre, folks, because crystallization is a property of chemicals. You usually don't think that cells will crystallize, right? So this result, for which he got the Nobel Prize, made people think, well, maybe the viruses are just made out of, say, protein, right? And then, in a repeat of the experiment, a British biochemist analyzed this crystallized tobacco mosaic pathogen and discovered that it was mostly protein, right, about 95% protein, but there was some nucleic acid present. So, in the late 1930s, the model of a virus was it was mostly protein with a little bit of nucleic acid, but nobody knew what they looked like yet. And again, folks, we just want to emphasize the reason our understanding of viruses lags so far behind our understanding of bacteria is viruses are just so much smaller, right? We couldn't visualize them using our compound light microscope. So for us, you guys, we're going to say that for most viruses, you need a much more powerful electron microscope to visualize the viruses, right? So the virus size range, um, we're down in the maybe 30 to 40 nanometers up to 300 to 400 nanometer size range. So the biggest viruses, for example, members of the pox virus family, we might just barely be able to see them with our light microscope. But in general, we're going to say most viruses we couldn't see with a light microscope. So our delay in understanding virus structure had to do with the fact we couldn't see them, we couldn't visualize them. And that was going to require the invention of the electron microscope. And this is so sad, you guys. So here we are in the 30s, working up to another World War. We're working our way up to World War II. My understanding is that it was a scientist in Germany that first invented the electron microscope. And then it wasn't until 1939 that the first electron microscope was either made or bought or brought to the United States, to RCA Company in New Jersey. And this was momentous because now there was the possibility of maybe visualizing what those bacteria viruses were. What were they? So there was a group of scientists that gathered um, when the electron microscope was first used to visualize a bacteriophage. And these are such cool bacteriophage folks, bacteria eaters. So they look like little lunar landers, right? So these microbiologists, and scientists were waiting with bated breath as the first electron micrograph was developed of the bacteriophage. And you can imagine the awe and just, disbelief as the image of this bacteriophage was developed. And this is a great quote, you guys. One of the scientists there, Professor Herr J.J. Brofenbrenner, said, "My God, they've got tails." So literally, my God, they have tails. So as humans, whenever we encounter something absolutely new, we try to connect it to something that we know already, right? So we can try to understand what it is. So um, Herr Dr. Brofenbrenner was correct. This is known as the phage tail, right? Other scientists describe them as being sperm-like or tadpole-like, right? And we will come back, you guys, in the next video and talk about bacteriophage structure. What we're seeing here is all made out of protein. So we got a protein head or capsid that protects the phage, in this case DNA. And then this portion down here is called the tail. This central cylinder here acts like a hypodermic syringe, and there's the equivalent of a hypodermic needle that the phage is going to inject into the cell wall and cell membrane of the bacterium to inject its DNA into it. And then these cool little appendages are called tail fibers, and at their tips will be special proteins called adhesins that will bind to very specific surface molecules on the surface of E. coli, for example. So I think, folks, what we'll do there is we'll stop this video. And we'll start here on the next video on the description of the early studies of bacterial viruses or bacteriophage by Max Delbruck, a fine playground for serious children to ask ambitious questions. So we'll stop here, folks, and then the next video will be on the structure and replication of bacteriophages.